Hi everyone, today in our discussion we will explain the insulin. The insulin, as you know, is a really important hormone because it plays a really important role in regulating the concentration of sugar or exactly the glucose in our blood. It acts as a key which unlocks the cell, allowing glucose to enter the cell and provide it with energy that it needs to maintain its life. Our subject today will be really important because it has a direct relationship with diabetes mellitus. And as you know everyone, that diabetes mellitus is one of the most common diseases affecting a human being. Even more than 10% of Iraqi people suffer from this disease. Insulin is synthesized in the beta cells, which are located in the central position of Langerhans islands of the pancreas, and it is secreted freely into the bloodstream. It has a half-life with only 6 minutes. Thanks God it's only 6 minutes, because if it is more than 6 minutes, it will cause a problem which is hypoglycemia. This was just a simple introduction for our subject. Now let's see the structure of the insulin. The insulin, as you are seeing here, is composed from two polypeptides. We have the polypeptide chain A and we have the polypeptide chain B. These two polypeptides are linked together by two disulfide bonds. These disulfide bonds link the cysteine amino acid in chain A with its brother cysteine amino acid in chain B. Now let's see the insulin chemistry and synthesis. Insulin synthesis starts with translation of insulin RNA by ribosomes attached to the endoplasmic reticulum. As you are seeing here, this is and forming the preproinsulin. The preproinsulin is composed from 4 parts. Here we have the signal sequence, as you are seeing here. We have the chain B polypeptide, we have the chain A polypeptide, and in between we have the connector peptide, what we call it C-peptide. This preproinsulin will undergo a cleavage in the endoplasmic reticulum, losing its signal sequence, as you are seeing here, forming a new structure, what we call it the proinsulin. The proinsulin, such as his brother, he will also undergo a cleavage in Golgi apparatus, forming the active insulin and the C-peptide, which will be finally secreted into the bloodstream. Now let's see the regulation of insulin secretion. There are many factors that stimulate the secretion of the insulin. One of the most important factors is the glucose itself, because beta cells are very sensitive to the glucose. So when the glucose is raised in our bloodstream, it will stimulate beta cells to release insulin into the bloodstream. Vice versa, if it is low, there will be no insulin into the bloodstream. So here we can recognize that any destruction in the beta cells will cause beta cells to not release any insulin, and that will cause type 1 diabetes mellitus. The other factor that stimulates the secretion of the insulin is the amino acid. When you are eating food rich with a protein, such as chicken or meat, of course the amino acid will be raised in your bloodstream, and that will stimulate beta cells to secrete insulin into the bloodstream. The other factor that stimulates the secretion of the insulin is the gastrointestinal hormones. Gastrointestinal hormones cause an anticipatory release for the insulin. What I mean by anticipatory release for the insulin is that the insulin will enter the bloodstream even before glucose enters the bloodstream, and it will be ready to metabolize it. I talked previously about the factors that stimulate the secretion of the insulin. So what about the factors that inhibit the secretion of the insulin? There are many factors that inhibit the secretion of the insulin. One of the most important factors is the epinephrine. So that's why we call it insulin antagonist. Just imagine with me, when you are feeling stressed, or if there's anything scaring you, such as a noisy dog running after you, of course you will need energy to face these problems. So how can you get this energy? You don't have to be worried, because our hero, the epinephrine, will solve the problem. It will stimulate liver to convert glycogen to glucose and release it into the bloodstream. And at the same time, epinephrine will inhibit beta cells from releasing any insulin into the bloodstream. So now let's see the effect of insulin on carbohydrate metabolism. Insulin will increase utilization of glucose for energy production in all tissues, because, as I said previously, that insulin acts as a key which unlocks the cell, allowing glucose to enter the cell and provide it with energy that it needs to maintain its life. So what about the excessive amount of the glucose? Shall we throw it away in the garbage? Of course no. Here the insulin will store it in many organs, such as the liver and the muscle and the adipose tissue. Insulin will increase glucose uptake by the liver by increasing glucose transporter type 2. And at the same time, insulin will prevent liver from releasing any glucose into the bloodstream by inhibiting glycogenolysis and gluconeogenesis. We can imagine that just such as a father giving money for his son, and at the same time preventing his son from spending this money. Such as that is the insulin. It will increase glucose uptake by the liver, and at the same time it will inhibit liver from releasing this glucose into the bloodstream. Insulin also will store the excessive amount of glucose in the muscle and adipose tissue by increasing glucose transporter type 4. The glucose will be stored as glycogen, which is the storage form of the glucose in the liver and muscle, while it be stored as fat in the adipose tissue. Here we can see the brain laughing on the other tissues. What's going on with him? Why is he laughing on the other tissues? He's laughing because he can receive his glucose without depending on the insulin. That's because the glucose can diffuse freely through the blood-brain barrier. Now let's see the effect of insulin on fat metabolism. As I said previously, that insulin will increase the glucose uptake by the liver by increasing glucose transporter type 2. Here, the glucose will be converted to very low density lipoprotein. This lipoprotein will be released into the bloodstream. Here, the insulin will degrade fatty acid from the lipoprotein and transport it to the adipose tissue by activating the lipoprotein lipase. And at the same time, insulin will increase the glucose uptake by the adipose tissue by increasing glucose transporter type 4. This glucose, after it enters the adipose tissue, it will be converted to glycerol 3-phosphate. It will combine with the fatty acid and form the triacylglycerol. Here, the insulin will inhibit hormone-sensitive lipase and trap this fatty acid and trap this fat in the adipose tissue. So here we can recognize why physicians always prevent diabetic patients from eating fat. That's because diabetic patients who suffer from insulin deficiency can't degrade fatty acid from the lipoprotein and transport it to the adipose tissue. So that will cause fat to accumulate in their vessels and cause atherosclerosis, which can develop and cause more severe cardiac attacks. This is all what I have in my subject. Our subject will be continued by my colleague, Nisreen. Thank you everyone for your listening.
After my colleague Haidar has discussed insulin, I'll be discussing something which has a complete opposite effect, which is glucagon. Now, glucagon is a polypeptide hormone secreted from the alpha cells of the pancreas. Just like insulin, it undergoes a number of proteolytic cleavages to transform it from a precursor molecule, which is a pre-proglucagon, into proglucagon and eventually into glucagon. Here you can see the structure of glucagon, which consists of 29 amino acids arranged in a single polypeptide chain. The mechanism of glucagon action. This pathway is known as glucagon signal transduction pathway. After glucagon has been secreted from the alpha cells, it binds with the G protein coupled receptors found in the cell membrane of hepatocytes. Now, on the other side of these receptors, we have the G protein. Initially, the G protein is inactive and has a GDP bound to it. But after this binding process, the GDP is replaced with GTP. This causes the activation of the G protein. Now, G protein consists of 3 subunits, alpha, beta, and gamma. After it's been activated, the alpha subunit detaches and activates adenyl cyclase. Adenyl cyclase then converts ATP into cyclic AMP. Here in this pathway, cyclic AMP is the secondary messenger, whereas glucagon was the primary messenger. Now, cyclic AMP activates protein kinase A, known as cyclic AMP-dependent protein kinase. Now, once it's been activated, protein kinase A has the ability to phosphorylate and therefore activate several target proteins. Here, the protein is glycogen phosphorylase kinase. After it's been activated, it in turn activates glycogen phosphorylase, which initiate the first steps in glycogen degradation, known as glycogenolysis. This entire pathway is done in order to raise the level of glucose in the blood. Regulation of glucagon secretion. We have several factors that affect how much glucagon is secreted into the blood. First is glucose, which is the most important one, since glucagon's main role is to raise the level of glucose in the blood. Once it detects lower levels of glucose, which is lower than normal of about 90 mg per 100 milliliter, the secretion of glucagon increases. And depending on how low glucose levels are, glucagon secretion could increase by several fold. Another factor is epinephrine. In the body, we have the hypothalamus, which acts as a radar to detect the levels of glucose. So once it detects low levels, such as in hypoglycemia, it promotes the secretion, it sends signals to the sympathetic nervous system to promote the release of epinephrine and norepinephrine. These then promote the secretion of more glucagon in order to raise the level of glucose. Finally, we have amino acids. Now, after a protein meal, the amount of amino acids in the blood increases. This causes the increase of secretion of insulin. And as previously mentioned, insulin acts on lowering the levels of glucose in the blood. So the body, in order to prevent the glucose levels from dropping too low, the body secretes glucagon. This is to maintain a stable level of glucose in the blood. Now we have its effects. First, its effect on carbohydrate metabolism. Now, as I mentioned, glucagon's role is to raise the level of glucose. Once they drop too low, the secretion of glucagon increases. After it's been secreted, it travels to the liver where it promotes the process glycogenolysis, as I showed you in the earlier pathway. Now, the enzyme glycogen phosphorylase catalyzes the conversion of glycogen into glucose 1-phosphate. Now, glucose 1-phosphate is then converted into glucose 6-phosphate by the action of the enzyme phosphoglucomutase. In the liver, we have another enzyme known as glucose 6-phosphatase. This enzyme acts on converting glucose 6-phosphate into free glucose, which then secreted into the blood in order to raise the levels of blood glucose. Second, we have its effects on lipid metabolism. Now, glucagon acts on activating the process lipolysis, which in the adipose tissue, which is the process by which triacylglycerol is converted into fatty acids and glycerol. Now, these two are taken up by the liver, where the fatty acids are oxidized into acetyl-CoA. The excess is used in the formation of ketone bodies. Ketone bodies provide energy for several tissues, such as the skeletal muscle and the brain. Glycerol is used in order to, which is also taken up by the liver, by the way, is also used in promoting gluconeogenesis, which is the process of making glucose in order to raise the level of glucose in the blood. Finally, its effects on protein metabolism. In the body, proteins are converted into amino acids by the process proteolysis. Now, once amino acids increase in the blood, the secretion of both insulin and glucagon increases. Insulin, however, takes amino acids into the skeletal muscle, whereas glucagon takes them into the liver, where it promotes gluconeogenesis in order to, and once again raises the level of glucose in the body. Finally, glucagon in high concentration has other effects, such as enhancing the strength of the heart, increasing blood flow, enhancing bile secretion, inhibits gastric acid secretion. However, all these have minimal importance in the normal body function. Now, as mentioned earlier, that glucagon opposes the effects of insulin. It shares this trait with others such as epinephrine, cortisol, and the growth hormone. These are known as the counter-regulatory hormones. Here you can see the processes by which insulin inhibits and glucagon increases, which are glycogenolysis, gluconeogenesis, ketogenesis, and lipolysis. In short, insulin inhibits all the activities that would result in the production of energy, whereas glucagon promotes these activities. And that's all I have about glucagon. I'll leave the rest to my partner, Marwa.
Good morning, everyone. Thanks, my cause Nesreen. Now I talk about other disease, hypoglycemia. Hypoglycemia is abnormally low level of glucose in your blood, less than 4 millimole per liter. When your blood glucose level is too low, your body does not have enough energy to carry out its activity. So, hypoglycemia is the most common associated with the diabetes. It's the mainly cause if someone with diabetes takes too much insulin. Now talk about the symptom of hypoglycemia. Symptom can be different depending on how your glucose level drop. So, it can be divided into 3 types. The first type, can you make feel hungry, increase heart rate, and like you want to vomit and the pale skin. Another type, moderate hypoglycemia. Often people may feel afraid and confused and nervous. A most dangerous type is the severe hypoglycemia. Also can cause coma or death and headache. Now about diagnosis of hypoglycemia. You will need blood test to check your blood glucose level. And have 2 types of the test. In the first one, the test include not eating and watching the symptom. Another type include the or another type of test include eating a meal that cause the symptom of hypoglycemia or low blood glucose level and lead to several hours. Okay. Now mention about why hypoglycemia can cause the last mentioned symptom. Inside your body, we have two mechanisms. The first mechanism by the adrenal gland. Inside your body, we have two mechanisms to retain the normal level of glucose. The first mechanism by the adrenal gland. Adrenal gland can release two hormones, cortisol and adrenaline. These two hormones stimulate the liver to convert its carbohydrate or its stored carbohydrate into the glucose and then release it into the bloodstream. And another mechanism by the pancreas. Pancreas also secretion another hormone called the glucagon. Glucagon also stimulates the liver to convert its stored carbohydrate and then release to the bloodstream. Also, hypoglycemia can affect the central nervous system. Now, have our last mentioned symptom can mainly rise of hypoglycemia can mainly rise from the neuroglycopenia. Neuroglycopenia is the medical term referred to decreased delivery or delivery of glucose in the brain, mainly due to the hypoglycemia. Glycopenia affected in the function of the neuron and also affected in the effects in the function and behavior of brain and also cause cerebral dysfunction or brain damage or death. Now continue with Tihad.
After my colleague Marwa, now we're going to talk about the most major insulin functional disorder, which is the diabetes mellitus. Now, the diabetes mellitus means that there's an impaired carbohydrate and fat and protein metabolism. And this one is caused by one of two abnormalities. The first is, all right, now to remember these two abnormalities, let's check out this picture. All right, now in this picture, we can see that there's a policeman. What the hell? Sorry. We can see that there's a policeman that's standing in the middle of the street in order to help those young students to cross the street and reach their school. All right, now this policeman are representing the insulin, and those young students are representing the glucose, and their school is representing the cells. All right, now in the first abnormality for diabetes mellitus is that there's a lack of insulin secretion, which means that there is no glucose entering into the cell. So as we can see here, that when there is no policeman, those young students could not cross the street and reach their school, as the glucose also cannot cross the plasma membrane and reach into the cell. Now this is known as type 1 diabetes mellitus. The second abnormality is that the insulin is pretty much normal, but the major problem is that there's something known as insulin resistance. Insulin resistance means that there's the cells will just resist the insulin. So the insulin cannot function properly and entering the glucose inside the cell. And now, this school here is closed. So even if there's a policeman which helped those young students to cross the street, they just still outside the school, which means that the glucose units are still outside the cells. Now this type is known as type 2 diabetes mellitus. Type 1 diabetes mellitus. Type 1 diabetes mellitus is caused by an autoimmune destruction of beta cell. This destruction is a result from two factors or two stimulus. The first is from the environment, just like a viral infection or bacteria or any other inducer. And the second is a genetic determinant. Now in case of genetic determinants, in normal people, the immune T cell are responsible to attack the antigen and leaving the body cell alone. This one is recognized or actually is controlled by the self-tolerance of the body. Now in the diabetic person, there is no self-tolerance of the body, which means that the T cell will start attacking the own body cell and recognizing them as a non-self and destroying them. And among these body cells are the beta cell, which finally will causing the lack of insulin secretion. Now this is why in case of identical twin or a zygotic twin, if one sibling develops the disease, the other will have the chance of 13 to 15% to develop it too. And this is actually true because one of the two factors, which is the genetic determinant, are already present. So it will be very, very easy for an environmental infection to develop the disease too. Now we have number of metabolic change in this type of diabetes. These are the hyperglycemia, the hypertriacylglycerolemia, and the ketosis or ketoacidosis. Now, once the glucose cannot entering into the cell because of the lack of the insulin, the cell will start suffering from the deficiency of glucose. And as in compensatory solution, the liver will start using the gluconeogenesis and the glycogenolysis to produce more amount of glucose in the blood. But as long as the glucose cannot entering into the cell, they will accumulate in the blood and causing the hyperglycemia. And this is the first metabolic change. Now, once the cells are suffering from the glucose deficiency, it means that there is no ATP production. So they will just start looking for other source of energy production, including the fat. And once the fat are now starting to mobilize, the hormone sensitive lipase activity will be increased. And this one is responsible for the degradation of the triacylglycerol from the adipose tissue. Now, this huge amount of triacylglycerol released in the blood will causing the high triacylglycerolemia, and it's also known as the high triglyceridemia, which is a very, very dangerous one. All right. Now, once the fat are breaking down into free fatty acid, they will transport it into the liver and convert it into ketone bodies. And the main rule for these ketone bodies is that they can enter into the cell and produce energy without any need of insulin. So as long as the cell will using the fat for production of energy, the ketone body will continue to release it into the blood and accumulating and causing the ketosis or ketoacidosis. Now, type 2 diabetes. This type of diabetes is much more common than type 1 diabetes. It's occurring about 19% of all cases of diabetes. And the main problem here, as we mentioned before, that the cell now resists to the insulin, which means that the insulin cannot function properly and entering the glucose into the cell. So as we can see here, in normal person, the insulin can bind to its receptor normally and trigger the entering of glucose into the cell. But in diabetic person, the receptor will change or alter its shape. So the insulin cannot bind to it any longer, and the glucose unit cannot enter into the cell no more. Now, this is known as insulin resistance. And as a compensatory solution for insulin resistance, the pancreas will start ordering the beta cell to produce more amount of insulin into the blood, and as a compensatory solution. But as long as the insulin cannot bind to its receptor, they will just accumulate in the blood and causing the high plasma insulin level, which is also known as an indicator for this type of diabetes. All right. But as long as the glucose cannot entering into the cell, these beta cell will continue to produce more amount of insulin until they become hypertrophied and tired and show lasting impaired function, as we can see here. Now, for the metabolic change for this type, they are just similar in type 1 diabetes, except that there is no ketosis, or also there is no ketoacidosis. And this is actually true, because even if there is insulin resistant, this few amounts of insulin have the ability to reduce the hepatic ketogenesis, which in turn reducing the ketone bodies level in the blood. This is why I have. And for more common complication for diabetes, this will be produced by my colleague Ammar.
Hello everybody, and after my colleague Ibtihal have talked about the theoretical aspect of diabetes, I'm going to talk about the clinical and practical aspect. And actually, it's really complicated to understand this aspect, so we're just going to get a general idea. And now let's start with diabetes symptoms. And it can include 4 symptoms: glycosuria, polyuria, polydipsia, and polyphagia. Now for the first 3 symptoms. As all we know, in normal person, glucose does not appear in urine. But when the concentration of glucose rises very high in the blood, as in diabetes, that it exceeds the capacity of kidney reabsorption, some of glucose will start to appear in urine, leading for the first symptom, which is glycosuria. And since glucose is an osmotically active compound, it will promote dragging of water with it and promote diuresis and excessive urination. Now this is the second symptom, polyuria. Excessive urination will lead for losing of body water and dehydration. This will lead for the person to feel thirsty. Now this is the third symptom, polydipsia, or excessive feeling of thirst. Now for the first symptom, in this picture, muscle and adipose cell look all sad and frown because glucose cannot enter these cells due to the lack of insulin seen in diabetes. Now, this will make these cells strive for energy, leading for lipolysis in adipose tissue or the breaking down of fats, and proteolysis in the muscle tissue or the breaking down of proteins. This will lead for weight loss and excessive feeling of hungriness, which is the fourth symptom, polyphagia. Now for diabetes complication, actually the complication of diabetes arises from the increased glucose concentration and hyperglycemia seen here, and it can be divided into two types: microvasculature, those affecting the tiny blood vessels, and macrovasculature, those affecting the large blood vessels. Now for the microvasculature, it can be divided into three types as well. The first one is retinopathy, affecting the retina of the eye, leading for what is called cotton wool spots, which are a whitish cotton-like spots on the retina, and also may lead for flare hemorrhages, which are hemorrhages in the retina of the eye. Now the second one is nephropathy, affecting the nephron. It actually affects the efferent arterioles and the glomerulus of the nephron, and may ultimately lead for nephrotic syndrome, which will by years make the patient in need for dialysis. Now the third symptom is neuropathy, affecting the nerves, and it can be divided into two types as well: peripheral neuropathy, affecting the sensation of pain and reflexes and so on, and autonomic neuropathy, affecting the function of the autonomic nervous system, like sweating, passing gases, and so on, and even the secretion of some hormones like epinephrine and norepinephrine. Now the macrovasculature complications can be divided into three types as well: coronary heart complication, cerebrovascular complication, and peripheral vasculature complication. Now a combination of both poor blood supply due to the vasculature complication and the nerve damage that will lead for the patient not feeling in pain may ultimately lead for severe ulceration on the diabetic foot without the person even knowing that, and may ultimately lead for the amputation and the cutting of the foot. Now let's see the way by which this complication arises. For the microvasculature complications, this arises from what is called hyaline arteriosclerosis, in which we have a hyaline deposit in the lining of these arterioles, making these blood vessels hard and inflexible. We may also see an increase in the thickness of the basement membrane in some of the capillaries, making it harder for oxygen to diffuse to body tissues and leading for hypoxia. Now the macrovasculature complication arises from the frequent development of atherosclerosis seen here, leading for heart attacks, strokes, and so on. Now let's see the pathophysiology or the mechanism by which this complication arises. Now as all we know, in diabetic patients, the concentration of glucose is rising in the blood and we have hyperglycemia. But the glucose cannot enter muscle and adipose tissues. But what about other tissues like the lens of the eye? Actually, glucose will enter these cells and is going to be converted via an enzyme called aldose reductase into sorbitol, which is an alcohol. The problem here is that sorbitol is both osmotically active and it cannot leave the cell, so it will promote the dragging of water into the cell and leading for some of the complication of diabetes like cataracts. Now other complication arises from the fact that excess glucose will start to bond to different body proteins in an uncontrollable way in a process called glycation, leading for disruption in the structure of these proteins. One example of glycated proteins is HbA1c that we are just going to talk about. Now for the diagnosis of diabetes. Diabetes is diagnosed by measuring the concentration of glucose in the blood naturally via different tests like fasting glucose test, in which the person does not eat and drink for 8 hours, and if he has a concentration of glucose from 110 to 125, this will indicate that he has pre-diabetes, and if he has more than that, this will indicate diabetes itself. Now other tests include random glucose test, oral glucose tolerance test, and so on. Now there's other way to indicate diabetes, which is measuring the concentration or the percentage of HbA1c in the blood, which is a glycated hemoglobin that has glucose attached to it, like we have just said, and it can be used to indicate glucose levels for the past 2 or 3 months. Now a percentage of HbA1c from 5.7 to 6.4 will indicate pre-diabetes, and if it's more than that, this will indicate diabetes, no doubt. Now this lecture was provided by the students of Medical University of Jaber bin Hayyan, hoping that it wasn't long or hard, and in the end, thank you.